I'm Shravan Kumar Reddy, and I'm here to present a survey on cancer classification using microarray gene expression data with deep learning. Coming to the abstract, we are here to develop and ensemble deep learning methods to overcome and the problems that the current patients are. We are here to develop an ensemble deep learning method which classifies the cancer, which is of 30 different types of cancers, and another one was normal type. So basically, there are many problems currently. The patients are facing many problems regarding the cancer types, and we are here to help somewhat what we can do for them by classifying what type of cancer it is after the prediction. Coming to the microarray technique, it is a multiplex lab on chip. Its purpose is to simultaneously detect the expansion of thousands of genes from a sample. And one of the major concern on this cancer classification part was there are many redundant genes and which do not come into effect while classifying the cancer. So we are trying to find out what exactly the genes that will be helpful for us, and what type of genes we have to consider to classify the cancer part. And our main objective is to develop a novel ensemble classification algorithm to improve the stability, efficiency, and prediction accuracy, and to remove the noise and irrelevant genes from the current data sets, like what we'll be taking from the patients and all that. Overall, to improve the computational cost also. And we have surveyed on reviewed many papers with reference to the IEEE papers. One of the first paper we have reviewed was correlation feature selection based improved binary BPS wall for gene selection and cancer classification. Coming to the main drawback of it was it gives result on only 7 data sets that have to check different. The main disadvantage of this was it was taking only a single type of cancer, and it was one algorithm was performing only for one type of cancer. So, and the most of the papers we covered through was having machine learning models, which the conclusion was to move on to the deep learning algorithms to get the better accuracy and the prediction parts. And yeah, the existing system and drawbacks was that the ML models with high dimensional data, the ML models are not that much accurate, and the suggestion was to move with deep learning algorithms so that we can get the accuracy more. And the proposal system was to develop a novel ensemble algorithm to improve the stability. We are currently in the implementation part. We have reviewed all the papers, and we have moved with the 1D CNN, 2D vanilla CNN, and 2D hybrid CNN with around 10,300 samples with 33 different types of cancers and 713 samples with normal tissues, which do not have any cancer. And with the current models, we have got 94.08% accuracy for 1D CNN and 94.95% with 2D CNN, and with 94.75 with 2D hybrid CNN. And we have used ensemble algorithm for that, and we got around 99.29% accuracy. The first ensemble one method was we have directly taken the sum, and second ensemble was we have taken the weighted sum. So we are currently improving this implementation, how do we get what type of genes will be effective, and to select the gene spot, and that's it. Thank you. Any questions?
Yeah. So I just wanted to know about one thing in your presentation. One suggestion is that you have mentioned drawbacks. Mention it as research gap.
Okay.
the main reason here why am I telling that word is, say it is drawback for when he written that title, it was his objective. So it is
Yes.
not called as drawbacks.
Yeah.
Whatever the persons or the research authors aim was, he would have done that. So it is
Yeah.
not a drawback. It is a research gap. That is, you are doing something in that particular new field, so it won't come a drawback. Use a proper word so that it is drawback means this shows that his paper won't is not up to good mark or something. That it looks a very bad image. So if you
Yes.
can mention and change the name, that would be great. And then what about the data collection? How did you collect these data?
So
For
we collected the data from the TCG, the Cancer Genome Atlas. Like it was a program. They're collecting the samples and they're placing
okay.
all things at one place to
Yeah.
make things
Okay.
easier.
Fine. I mean pre data which was available in net, you just copied it.
Yes. Yes.
Okay. Okay. And what was the main objective of your research? Because you have already taken the current data which is already there, and you're doing the analysis. What is that? How are you? What speciality of it of your research? I
Yes.
couldn't able to judge it.
So we are trying to uh, like develop an ensemble algorithm with deep learning models so that we get the more classification. Like it will be more accurate than the previous ones, so that like the error will be less and the patients will get the accurate results. And to get that, like we are trying to select the best genes that will affect the classification of the cancer.
Okay. Okay. Uh-huh. So, but the datas are not from. It's already available data. You didn't collect any data which are there, right?
We are currently like completed the review paper, and like in the first phase, like to get the what will be what exactly the results are we are getting, we have taken the sample data from there, like this we will continue this to get more sample data from any other like any other some hospitals or something from there, and we will complete this.
Okay. And one more thing I just wanted to understand here is with respect to the um, like uh, uh, okay, like augmented reality you mentioned in your title, right?
Sorry.
What's the title? Can you just deep learning? Fine, fine.
Yeah.
Just the data. Yeah, 22, right? Your serial number.
Yes, sir. 22.
Okay, okay. So classification of deep learning. So, what is the gene expression date? Last part of the title, microarray gene expression data. Like, yeah.
Sir,
Can you just throw light on that?
yes, sir. So it will be the data that we are getting from the already research papers. Like it will be the gene expression, like what exactly the gene we are getting. Like there are already some pre-processed methods on genes, like exactly like some there will be number of genes, and we will get the like some we will give assign some number to one type of genes, so that like with respect to some parameters what they have considered, like there are so many things, and like it will they have processed it with the images like microarray from.
Okay. So, what would be the implementation cost or the next future of this project?
As we just started on the implementation phase, like we are trying like how efficient, like how fastly we can run that algorithm, and how efficiently we will get the like what type of genes to select, and exactly like how fastly we can predict the algorithm, like the cancer.
Which stage of cancer the data which you have selected?
Sir, exactly. Currently, it has like it will tell not we are not go.
What type of cancer?
Yeah, what type of cancer it was, and we can improve that to like at which level the cancer was.
No, I asked which type of cancer you are talking about.
So we have taken 33 different types of cancers.
All the different types of cancers you can rectify, is it?
Yes, sir. The algorithm, while we have given the data, it predicts like any of these 33 type of cancers, it will detect, it will classify it.
Okay. In implementation part, what are you actually looking for? Because cancer is basically, we all understand it is just a one extra body tissue which has been not required, which has been, which is being growing. That is what as per our knowledge, right? So
Yes.
what exactly, I mean, whichever part it grows, that part we just call it as a extra tissue, or which may lead to some failures of organs, or rather, I mean, complexities of biological will be there accordingly. So the
Yes.
point here is, what type of implementations you're planning to make with respect to this, like how you can say for example blood cancer, or breast cancer, or leg cancers will be there, or different types of cancer, like mouth cancer or something. Many different types of cancer are there. What is that particular type of cancer you're talking about?
Sir, while we have taken like from the genes expression, we have considered lung cancer and we have taken consider into kidney cancer and colon cancer. So there are 33 different types. Like we have considered like it is fine difficult to like to mention about one. Like where we can get the exactly the gene expression, we can give input to the model and we can get the type of we can check on the type of cancer.
Okay, fine. Thank you.
Thanks, sir.
For your suggestion.